So when you're finally ready to do your simulation with the map here and collect your data, and you open the PowerPoint simulation, you're going to want to just keep it in edit mode. Don't go and press the slideshow, because then you won't be able to manipulate the different items that are on there. And so from here is where you'll do your work. And if you highlight something and you zoom in, it'll zoom into that thing you've highlighted. So for example, if I had pasted my data here, not the data, but the coordinates that we're the random locations that we're gonna sample from. In the earlier video, we saw how to do that with Excel. But since I'm highlighting there, if I went and zoomed in, it's gonna take me to that spot there. So that's just something to consider. Wherever you want to zoom in, just highlight that object. Another thing is you can make what you're looking at, give yourself more space by getting rid of this menu ribbon here. So there you would go to the right here where the arrow is, and then just get it out of the way. If you needed that ribbon again, you can just click on here and it comes back down. But you don't want it to be pinned there. So let's just get it out of the way, and we're ready to start. So the first coordinates that we're going to collect data from are 1 meter east and 28 north. So this vertical red line is going to help me find the 1 meter. The scale there is on the bottom for running east, and then you can use your arrows to move the lines a little better. And then we're going to go 28 north. So this is 25, and there's 28. And so now we're going to take our transect here, which is a 10 meter transect with 1 meter intervals. And we're going to move it as best we can to where the left end of that transect lines up right where the two cross. And now we can zoom in more closely, a lot more close if you want. And we can fine tune with the arrows and make those adjustments. And now I can move those guidelines out of the way. And now I can see my transect's been laid out at a random location, and I'm ready to collect the frequency, number, and coverage, which was described in the last video. Another thing you might do if you want to have objective value for how open the habitat is, how much bare area there is, you can also record the length of the transect that it has no coverage. But that's it. You wouldn't do frequency because it doesn't make sense to do frequency or number because those are values for plants that are touching the transect. So you can see there's a lot of the transect that's not being touched. And here you're only considering the horizontal line. Those little vertical lines, little marks are just marking where each meter would be, each meter interval here. So when I scan that horizontal line, I only see two different individual plants touching it, and I'm going to move it out of the way real quick because I had a hard time seeing that letter. So that's a C and that's a T. And actually T is the little dot right in there. The T, some of the T's are pretty small individual plants. You can see that in some of the other areas there. There's a C here on the outside, so the larger circle is C, the small dot is T, and then there's an S right there. And these are separate species. There's a key on the bottom what the names of the species are, but for now let's just write down letters. So there's your C there, and the larger C, so really we're only getting C in this one. And since I moved my transect out of the way in this simulator, I can get it right back by just hitting Control-Z, and it goes back without me having to put those red guidelines for us here. So when I look across here, keep in mind that first large circle is just C. So for the first one we're going to record is C, and I have my data sheet here that I'm going to write in. And so I'm going to write C as the first one. Now if I'm keeping track of the bare ground there, I might start with that one, and I would just put U for uncovered or some letter, or just bare, the name bare on there. And so for C, the first thing I'm going to do is frequency, and that's going to be the number of intervals that it appears in. So I see it here in the first, not the first interval, but the second one is there, and then a little bit of it goes into the second one. So that's 2 intervals, and then the next individual touches there and there, so that's 2 more intervals. So this is a total of 4 intervals, so I would write a 4, and then I'm going to put a comma after that in my data table because I'm going to do this transect, 9 more transects, and I could come across species C again, probably will, as chances go. Now for the next column, the easy one is just the number of individuals. There's 2 on the total transect. And now I have to get the coverage, the total coverage. And here's where you may have to do a little bit of adding up. You might do it in your head, or you might do it on a scratch paper. Remember that each line is an interval. Okay, so, and the best we can do is, in our minds, is divide them up into tenths. So if I look in this first interval here, where C is occurring, it looks like maybe it covers about 0.8, and then in the next one it goes about 0.2. So 0.8 and 0.2, in my mind that's 1. And then in this next one I see another 0.3, so that's 1.3. And then going into the next one, it looks like another 0.7 or 0.8. So 1.3 and 0.7 would be 2. If we thought it was 0.8, it would be 2.1. So I'm going to put 2 or 2.1 in the last column for coverage. Okay, and then that's it. Now if we were keeping track of bare ground, there's like 1 meter, 2, 3, 4, 5, 5.2, another 0.8, so that's 6, 6, and then there's another 6.8, and then there's one hole in there. It's about so I lost track of what it was with my thoughts. So it's 1, 2, 3, 4, 5.2, another 0.8, that's 6, 6.8, 7.8, about 8 meters. So that would be about 8 meters of uncovered ground there. And so you're done with that transect. And so now you move on to the next transect. So far in my field notebook, I have bare ground. I'm going to keep track of bare ground. So we can't record frequency or number of touches there, but there was 8.0 meters of bare ground. It's desert habitats, mostly open. And we only had 1 species in that first transect. And in that first transect, we had a frequency of 4 intervals, 2 individuals altogether, and 2.1, and I put a comma after that. So this would normally be in my handwriting, and it is on my little notepad, and then I just transferred it here to show you what my field notes actually look like. I would have had a data table and then wrote these numbers in there. So now I'm going to move on to the second transect. And so for the second transect, I'm probably going to want to zoom out so I can see a little better. I also need to come back here and check what my next coordinate is. My next coordinate is 8 and 31. So I'm going to have to move my vertical line 8 meters east, and it was 31 north. So this is in the way here. So 30, there's 31, and fine tune a little. That looks about right. So 8 and 31, and then I'm going to move my transect there, and we've established that transect. And I'm going to zoom in now since I'm highlighted on that little tool we're using to simulate a transect, and it'll take me right to that area there. And I'm going to adjust it to where we meet right at the corner where the 2 meet, the 2 guidelines. I'm going to move them out of the way so I can see better. And boy, this is tough. So let's highlight and zoom in a little more. And it's a tough call, but it looks like S may or may not be touching right there. I don't think it is. It looks like we're running through D, and we got to be careful here because there's a little T in there, and it seems that the larger one is missing a letter there. But because this is creosote dominant habitat, which I happen to know based on the description already, we don't always know that. We go in there, we get a feeling that the creosote bush is dominating the area, but we want hard numbers. We want real numbers to support that idea there based on just walking through there. So we're going to call this last one a CNT, which tends to be a smaller species in terms of how much coverage the plant itself covers. is that little one there. There's overlap. So creosote's actually it's a bigger shrubby type, and then tea may be a smaller plant growing underneath it, which means it may be a little more tolerant of shade. And again, if you want to move this out of the way so you can check the letters a little more closely, you can move it. Just do a solid move, and then look. And I say, I know that's a C. This is an E, and then we have another C, which is with the larger one. And you can get it right back by hitting Control Z, and it takes you exactly back to the same spot. You can see that we're still not touching S there. So on my data sheet, as I'm going there, S is not touching. There is nothing all along here. So I'm going to do my bare ground coverage real quick. So that's 1 meter, 2, 3 meters, and then about 0.8, so that's 3.8, and then I see about 0.4 more, so that's 4.2, 5.2, 6.2, 7.2 to that line there, 7.2, and about 0.6 more, 0.6, so that's 7.8, and then just this little part right here, about 0.2 more. So that's about 8 meters again. That's the bear. Now we're going to look. The first one that we come across here is E, and I don't have that on my list, so I'm going to write that down. Species E. Again, the key for that is on the bottom. And then for E, I don't see E anywhere else. Remember, this one is a C here. So I'm going to go ahead and label that as C. And so all I see is one E. So, and we're going to look at the frequency of intervals. It's in this interval right here. Oops, let me move that back. It's in this one and this one, so that's 2 intervals only. There is no other individuals there. There's only one individual. That's number 1 under the N column. And then the amount of coverage there. So here it looks like that's about 0.2 of the 1 meter interval, so 0.2, and then on this side here, it looks like about 0.6, so I'm going to call that 0.6, and 0.2 is 0.8 meters, okay? And then I'm going to put a comma after all those values. The next one, we come across one individual of D. There's no other D's on here, and it's only in one interval. It's between this line right here and this one. Between this line and this one here, and so it's only in one interval, so I'm going to add D to my list, and that's going to be a 1 interval. There is 1 individual, and it covers, I don't think it's right exactly halfway, so I think it's 0.4 meters. And then we have 1 more touching here. Now, this whole big circle here represents 1 individual C, which is creosote probably, but it only touches in this transect. The transect ends right there, right around where that little T is, and the T is not touching the line, horizontal line. So this large C individual is only in 1 interval. So I'm going to go back to where I wrote C back in my first transect, and I'm going to put a 1 after the comma. And then there's only 1 individual, so I'm going to put a 1 in the second column after the comma. And then I'm going to add how much it covers, and it doesn't cover the whole meter. It looks like it's about 0.8, so I'm going to put 0.8 after the comma in the last column for C. So here's what my raw field data looks like so far. You can see that for the bare ground, I added a comma and 8.0 more meters of bare ground. For C now, the row with C has a second value in each one because we saw C again in the second transect. So we had a frequency of 1 individual, and it covered only 0.8 meters. And so now we're going to have to total these when we're done with all 10 transects. We've done 2 so far. And then we added 2 new species on our second transect, and we may run across them again. That's why we put a comma there. So you can see we had a frequency of 2 for E and 1 for D on that second transect, and then there was only 1 individual of each. And then E had only covered 0.8 meters, and D covered 0.4 meters. So now in this video, we'll go to our third location, which the third transect would be at 23 and 6. So I'm going to take my east mark and go to 23. There's the 20 mark on the bottom, 21, 22, 23. And then it's 6 here, so this is going to come 6 north, so that would be about right there. Down there. And so this would be setting up the third transect at that random location. Again, you zoom in and fine tune it. And now you just go and record the data further. So now what I'm going to do is cut this video off, and I'm going to start collecting data here. You should try to practice collecting this data yourself, and then the next tutorial will show how to analyze that data. So I'm going to go ahead and pause the video for now, and then continue collecting the data. It'll go a lot faster if you're not talking and trying to explain it all the way through. Once you get the hang of it, it'll go a lot faster. So that the video will cut off here so you can get on and practice collecting your own data so you get an understanding of where the data is coming from. So after running the remaining transects and collecting the data, this is what the data looks like. I got a total of 9 species, and those are the numbers that we're seeing. For the bare ground, I think it totaled about 86 meters out of the 10 intervals and 10 transects. It was total 100 meters, so that would suggest a lot of open space, which is what the map looks like. Now all that's left is to go in here and to add up the numbers in each box so that you get a total frequency for creosote, a total number of individuals, and the total amount of coverage for each box. And then that data actually goes in the table 20.1 from your lab manual for the final analysis, which will tell us what the more dominant species are or importance values. So here's what the totals look like, 86.9. Some cells had only one observation for like species I, a frequency of 2, one individual, and only 0.9 meters, but the totals are in there. And it's those totals for each box is going to, what's actually should go in that table, the summary of it, and then from there we run the calculations. So here's what the totals for each box would look like, and these data now are going to be analyzed to figure out from these individual frequencies and numbers, individuals and coverage, we're going to figure out the relative frequency, relative number, or relative density, they call it, and then the relative coverage. And so the next tutorial video will show you how to do that.